Right. Right. So we can start now 9:30. Uh, right. So this lecture is on actually these are two lectures on gastric secretion and gastric motility. Right. So physiological aspect of main function of the stomach, that is secretions. Of course, it helps to digestion and the, some sort of motility function. So these are the ILOs, and so within these two lectures, I will cover these ILOs and some of these things like acid secretion and gastritis and acid secretion related problem, mucosal barrier. You will learn in the tutorial as well, right? So relevance of stomach or gastric physiology, I would say huge, especially gastritis, right? Gastritis is very common, right? I'm sure some of you, considerable number of you, had a gastritis at least once in your life, right? So gastritis, so gastric peptic ulcers are common. And as I discussed in the previous lecture, reflux disease, gastroesophageal reflux disease is related to stomach for the acid, common. And nowadays, removal of stomach also getting common as a surgical procedure, especially for to cosmetic or reduce the weight. Removal of part of the stomach is getting popular even in uh, not only Colombo, like other hospital, Kandy and Karapiti and those places also. Now they will do the removal of stomach to reduce the food intake. That's the only purpose, and reduce the weight, right? So that is also getting relevant nowadays. Right. So gastric secretion, we have huge secretions, large volume of secretion inside the stomach, right? So it's say about 2.5 liters of secretion per day, right? It's even probably more than the urine output, right? So probably urine output also somewhat closer to that. So it's a huge secretion will take place in the stomach, but most of them will reabsorb. So that is why we will not lose this volume of fluid. Right. So basically two types of tubular gland, gastric gland and pyloric gland can be seen there. You will learn in the anatomy these structures. Gastric gland mainly upper part of the stomach or fundus. Pyloric gland in the lower part or antral portion of the stomach. And like this, these are the glands. So inside the glands, we have cells. As example, parietal cells or oxyntic or chief cells. We can see inside the gland. Right. So gastric gland, that's the main type of gland. 80% of glands are gastric gland. Contain three types of cell. Neck cells mainly secrete mucus, and then peptic cells. That is very important for the production of pepsinogen and gastrin, or HCl. Sorry, gastric lipase. Then parietal cells, oxyntic cells, the cells that produce HCl acid and intrinsic factor cells. So peptic cells and parietal cells are two important cells. In the histology also you will learn. Then other type of gland in the lower down close to the lower end of the stomach is pyloric gland, mainly secrete mucus. And of course, some hormone like gastrin will secrete from some of the cells there. And so you can see the main cells, main gland, gastric gland, or the gland in the upper part will have peptic cells and parietal cells. Then, of course, these are two types of gland. But in the surface, mucosal cells, in the surface, we have a lot of secretory cells, mucosal cells. Secrete mucus, bicarbonate, trefoil peptide, like these three things important to produce the protective barrier that protect the cell layer of the stomach, or mucous cell layer or inner cell layer of the stomach, because acid might damage. So surface cell produce these mucus, bicarbonate and trefoil peptide, and produce mucosal layer inside the stomach, and which will prevent acid contacting with the mucosal cell. With the acid come to contact with the cells might damage, right? So regulation of gastric secretion, and that is the one of the main physiological relevant area, regulation of the gastric secretion, right? So we can say three type of regulation. It's cephalic influence, gastric influence, and interstitial influence. So we can, for the educational purpose, to learn, right? Only to learn. We divide them into three phases, so three influences: cephalic, gastric, and intestinal influence. So to start very briefly, three type of influence. So let's say we have stomach like this, right? So we are talking about the gastric secretion, or basically secretion of the stomach. Right? When it's a gastric secretion, secretion of stomach, including acid and other secretion, right? In the cephalic influence, what happened through the nerves, mainly through the vagus nerve, coming to the through the stomach, we can initiate the secretion, right? So this, of course, this will occur as a reflex manner. To So this is the efferent of the reflex arm, right? So these are coming from brain. Let's say we have brain somewhere here, right? So these nerves coming from brain, right? So to get afferent or stimulate, get some sort of input to the brain, we have nose for the smell, eye when we see the food, or even thinking. So these factors will send impulse to the brain from eye, from nose, right? So the Darunahama nose, right? Or even thinking will send impulse to the brain, and there's a regulatory center somewhere here, and through that center, impulse will go to the stomach and initiate the secretion. So basically, I'm trying to say, when we see the food, when we get the smell, or when we think about the food, this system will operate, this reflex will activate, and through the brain, send impulse to the vagus nerve, that is mainly main parasympathetic nerve to the stomach, and increase the secretion, initiate the secretion. Since it operates through the brain, we call it a cephalic influence. Cephalic means basically related to brain, right? So it's operate through the cephalic, through the brain. And these reflexes are, we call conditional reflex, because we can condition them. Probably you remember, or you might learn something called Pavlov's dog experiments in advanced level. මතකද එහෙම එකක් කරා? Do you remember anybody? We first ask, give dog, we give food to the dog, and then dogs, of course, dog will eat, and then we will do that. After some time, we ring a bell. When we give the food, we ring a bell. Of course, in that experiment, somebody dissected stomach of the dog and put a tube out and measure the secretion, gastric secretion. So when dog get the food, gastric secretion initiate. But later we introduce food and bell. Right? And secretion increase. But later, without giving food, just ringing the bell will initiate the gastric secretion. Did you learn it somewhere? Hmm? That is how somebody discover the conditional reflex. Have you heard about this experiment earlier? Called Pavlov's dog experiment. Yes or no? කතා කරන්නේ no no response. අහලා තියෙනවද නැද්ද එහෙම එකක්?
No, sir. No.
නෑ?
Nah.
advanced ඉස්කෝලේ කාලෙවත් එහෙම එකක් අහලා නැහැ. fabulous dog response. Right, maybe now it's not in but in our days it was in the advanced level or O level books. Right. So that is exactly the same thing. This reflex, right? So when we see the food, when we get the smell, when we think about the food, right? Gastric secretion initiate. So same thing that I mentioned about the last lecture about the salivation. We get the salivation when we see the food, when we think about the food, right? Even with the smell, uh, we get salivation. So that will operate as a reflex or cephalic reflex or reflex operate through the brain, right? And afferents coming from eye or nose go to somewhere in the brain, and then vagus nerve, that is parasympathetic, will come to the stomach and do the secretion. So that is cephalic influence. So you can see now, even before food reach the stomach, or even before food reach the mouth, this reflex can operate. This process of secretion, gastric secretion, will operate, right, through the we call cephalic influence, through this reflex arc. Then we get the food, and now food came to mouth, right? So now mouth and then esophagus, and now food bolus is in the stomach, right? So let's say this is food bolus, right? So when the food bolus come to the stomach, this food bolus might stimulate local receptor. So let's say we have receptors local in the stomach, right? Some of them are mechanoreceptor. That means just touching the food, this touching the receptor, that will stimulate. Or maybe chemical receptor, right? So somehow this will initiate local reflex. So let's say this is a receptor, and this receptor will act on some neuronal flexes in the wall of the stomach that I mentioned in the first lecture. Local reflex can be operated through the nerve flexes, and that is afferent, and then efferent will come from there and do the secretion. So that is the local reflex. Of course, we can call it a gastric influence. So it will operate locally without connected to brain or spinal cord, right? So for this local reflex, mechanoreceptor and some nerves and receptor as well as hormones are important. Gastrin is a one main hormone. When this mechanoreceptor, or when the food is there, if it is touch the receptor, it will stimulate secretion of gastrin hormone. Gastrin is a hormone, and then gastrin hormone will activate and increase the secretion of HCl and some of these secretion. So considering volume of secretion or amount of secretion, this is the main influence, right? So that means more than the cephalic influence, gastric influence will operate in a higher level to increase the gastric secretion. So this one is first one, and this one, gastric influence or local reflex, is the second one. But the second one is the most potent, prominent, volume wise. It can create large secretion, right? Then the last one, let's say food is now in the stomach, get secretion, digestion is started, and some sort of breakdown to particles started, and so now it's almost done. Right? Job of the stomach is done. Now is food moving down to the duodenum, right? So food is moving to the intestine. So when the food is moving to the intestine, what should happen to the gastric secretion? මොකද වෙන්න ඕනේ? Let's say acid secretion. Now food is moving down. Yeah. Gastric secretion or gastric acid secretion. Now what should happen?
Should be reduced. Sir.
Yes, should inhibit, should reduce. Because now if the acid secretion or gastric secretion continue, that might harm the stomach, that might digest or damage the wall. So now it inhibit the gastric secretion or gastric acid secretion. When I say gastric secretion, it include the acid and other secretion, right? So when the food bolus coming down, what happen? Some receptor or some receptor mechanism operate and secrete certain hormone from duodenum or small intestine. So this hormone come and act and inhibit the gastric secretion. So it's a inhibition. It's negative inhibition. So that is the third influence, right? Intestinal influence, because it's coming down and mainly it's hormone, right? Gastric inhibitory hormone, GIP, somatostatin, and so on. So some hormone coming down and act on the stomach or gastric cells, gastric mucosa, and inhibit the secretion. So it's a mainly inhibitor, right? So now you can see broadly, very broadly, three type of regulatory process, right? Through the brain, we call cephalic influence. When you see the food over here or smell, or even when we think about the food, this will operate through the vagus nerve. So this will come through the parasympathetic and vagus nerve. Gastric influence is locally operated. Receptor, neuron, hormone will contribute to the local operation. Then from the lower down, some hormone secrete from the lower part of the lower part or lower area, or that means intestine, will inhibit the gastric secretion. That's the inhibitory influence, right? So this one is the gastric influence is the volume wise. That's the major contributor for the gastric secretion, right? So I just explained three phases or three mechanisms, and that will operate to regulate the gastric secretion, right? So cephalic influence, I already mentioned, food intake, see, smell, imaging lead to reflex secretion. Even low glucose level might trigger this reflex. අපිට බඩගිනි sugar glucose level එක අඩුයි, that might initiate reflex. So Pavlov's dog experiment, you can read or search in the internet. So vagus is the main nerve, carry that. Amount secreted is limited compared to the gastric influence. Gastric influence, that is the local gastric factor influence, quantitatively most significant influence. And something I forget to mention, for the gastric influence, I already mentioned it's a local operated. So let's say this is stomach, and I mentioned that it's a locally operated. So receptor is here and stimulate, and in the wall, probably stomach wall, some nerve flexes and effector that will act on the secrete cells. So that's a local reflex operate in the gastric influence. So other than that, for the gastric influence, that means when the food particles come to the stomach, some central reflex also will operate. For that central reflex, this receptor, let's say there is a receptor here, will touch the food bolus, will send impulse to the brain. through the vagus nerve. And from the brain, impulse coming to the secretory cells through the vagus nerve. So both afferents and efferents will go through the vagus nerve to the brain. Don't confuse, it's not cephalic influence. We will not put it to the cephalic influence because input is also from the stomach. For the cephalic influence, input or afferent coming from eye, nose, brain itself, so on. But here, afferent will go from the stomach and will operate through the brain. So therefore, this reflex we call vagal reflex, because afferent also go through the vagus nerve, efferent also will come through the vagus nerve. So we call vagal reflex. That also important to for the secretion and for the motor function of the stomach. This vagal, මේකත් vegas, යන්නෙත් vegas එකෙන් එන්නෙත් vegas. Afferent also vagus nerve, efferent also vagus nerve, but operate of course in somewhere in the brain. So it's not really local reflex. This one is local reflex, this one is central reflex, but still we categorize them to the gastric influence, right? So that is important for the gastric secretion. And of course, as I mentioned earlier, acid secretion can be inhibited by the local reflex also, other than the reflex activate through the lower part of intestinal influence, through the gastric reflex. This local reflex will inhibit the acid secretion also. As example, when we digest the food and when we get adequate acid, excess acid secretion might be harmful. So local reflex will operate this manner and inhibit the acid secretion, that also under the gastric influence, right? So gastric influence is the main influence for the acid secretion, which will operate to the locally as well as through the central reflex. Then, as I mentioned, intestinal influence operate through the hormones secreted from intestine. Secretin and GIP, gastric inhibitory peptides, are important hormone for the inhibition of gastric secretion, and as well as they are important for the inhibiting the gastric emptying. You know, when the here I am talking about only secretion, but when the food content come to the stomach, it should gradually empty to the duodenum, right? So this, especially GIP, inhibit the gastric secretion as well as gastric emptying. So therefore, some GIP function, gastric inhibitory peptide functions, will use to treat the diabetes mellitus. මොකද gastric emptying එකනේ මේකෙන් කරන්නේ. මේ gastric emptying, if we can actually inhibiting the gastric emptying. So this is the inhibitory hormone, secretin and GIP inhibit the gastric secretion and gastric emptying, right? So if we can delay the gastric emptying, we can prevent increased blood sugar level. Emptying prevent කරනවා කියන්නේ වැඩි වෙලාවක් content will retain in the stomach, right? So that will helpful in the diabetes patient, right? එකපාරට ගොඩක්, if the large volume of glucose absorbed into the body, blood glucose level will increase. So delaying gastric emptying or delaying movement of food or chyme to the small intestine might help in the diabetes patient. So some drugs will increase GIP, gastric inhibitory peptide, or action of gastric inhibitory peptide, and will help in diabetes mellitus patient. As example, there is a drug called sitagliptin, that will help, right? So this is intestinal influence, right? So later, try to write briefly to this question: what are the main regulatory mechanism of gastric secretion? You need to mention 3 phases and how does it operate. Right. Now come to the acid secreted by parietal cells. So as I mentioned earlier, parietal cells is the cells that secrete acid, HCl, right? So the parietal cells, yeah, this is parietal cells. You can see this is parietal cells, and this is the lumen, and this is the blood, and this is the cell. This is interstitial space. Of course, it contains blood vessels, and this is the lumen. So in the parietal cells, you can see H ion and chloride ion secreted by a separate mechanism. Of course, then they can bind and make HCl, right? Uh, Secretion of H ion is active ATP dependent process. Therefore, we call proton pump. What is proton? What a proton can? You learn in that answer. What is proton? Huh? Nidida. What is proton?
H plus.
Yes, H plus. Other name for H plus is proton, right? So therefore, we call this is H plus pump or proton pump. It needs ATP. So H plus secreted in this mechanism, and chloride ion come to a passive process, and this is active process, need ATP and get the HCl, right? So this is what happened in the parietal cells. So to control this process in the parietal cells, we have 3 receptors. So let's say this is the parietal cells and secrete HCl. It has 3 receptors. M receptor, right? M receptor, actually acetylcholine secreted from vagus nerve will act on M receptor, right? So then acetylcholine secreted from vagus nerve will stimulate the M receptor or activate something, right? So M3 receptor or M receptor is receptor for acetylcholine, which is secreted from vagus nerve. You can see it's vagus nerve, right? So that is how vagal influence or cephalic influence both operate through the vagus nerve, increase the secretion of acid in the parietal cells. Then we have some other receptor we call H2 receptor and substance called histamine. I'm sure you learned histamine in the previous the blood module. Even in the foundation module, I think you learn histamine, and histamine will act on H2 receptor. So that's the second receptor. And third receptor, we have CCK receptor that we call, that is the third receptor. First one, second one, and third receptor, CCK receptor. So hormone gastrin will act on CCK receptor. So it's a hormone mediated process, right? As you can understand, this is the nerve mediated process. Acetylcholine is a neurotransmitter, and this one is not really hormone. Histamine is not hormone, but we can call it, it's a substance. So somehow these 3 receptors in the parietal cells will stimulate the acid secretion. So if there's a stimulation for the acid secretion, there should be inhibition also, right? So I will explain later. So acid secretion can be inhibited as well as secreted through these receptors, right? Right. Right. So these are the 3 receptors: acetylcholine, CCK, right? So acetylcholine act on M3 receptor, gastrin will act on CCK receptor, histamine will act on H2 receptor. So these are the 3 main receptor which regulate the acid secretion. It say there's a synergism, some sort of high rate of secretion can be stimulated with relatively small change in the availability of each stimuli. It's like when 2 stimuli try to stimulate these parietal cells, that will get the additional effect than the just a plus effect. Right? දෙක එකතු කරලා එකතු වුණාම එන effect එකට වඩා වැඩි effect එකක් එනවා when two receptor will stimulate, that will call synergism. Right. So right. So I will explain this this activation of these three receptor, and this is something you need to understand with you need good attention for this. So just get a short break. වතුර ටිකක් බොන්න ඕන බොන්න. So because are you in the where are you at home? Not at home. කොහෙද දැන් ඉන්නේ ඔයගොල්ලෝ? මොරටුවේද මාළඹේද මේ වෙලාවෙ ඉන්නේ? ඔය ගෙදරද?
So we are at NSTH.
So where? අවුන්නේ කියපු එක.
NSTH, sir.
Okay. So it's මාළඹේ. Elephant. Right. Okay. Everybody, I mean, are you in the lecture theaters? Are you in a lecture theater or in anywhere? I mean, you can sit somewhere if you want. එහෙමද දැන් ඉන්නේ? The lecture එකකදී වාඩි වෙලා ඉන්නේ.
No, sir. We are in the study area.
Study area. Good. Do you have Wi-Fi? Wi-Fi තියෙනවද?
No, sir.
Data.
Yeah.
Okay. Right. Right. Okay. So if you want, you can go and have a cup of water. මේ නිදිමත නම් ටිකක් අතපය දිගාරලා වාඩි වෙලා නැගිටින්න. Because this is something you need to understand, not to memorize. හරිද? Right. So, right. So this is the parietal cells that I mentioned that will secrete the H plus or secrete the HCl, right? So as I mentioned early, acetylcholine coming through the vagus nerve, this is vagus nerve, can stimulate it. Then I already mentioned gastrin hormone. This is the gastrin hormone will come and act on parietal cells. So gastrin is a purely a hormone. So at the first lecture I mentioned we have some hormone and some substance. They are not real hormone. We call it they act in the paracrine fashion, and they just locally diffuse and do the job. But gastrin, it's not that. Gastrin is a purely hormone, right? If it is purely a hormone, how does it come to the parietal cells? Parietal cell එකට එන්නේ කොහොමද? Hormone එකක් නම්.
Through the blood.
Through the blood. Yes, very good. So as you know, it's a hormone, endocrine hormone, antara sarga paddhatiya කියලා කියනවනේ in advanced level. So they will come through the blood. So gastrin actually secreted from the cells in the stomach, somewhere in the stomach, lower part of the stomach, but they will enter to the blood vessels. You can see circulation, and through the blood vessels, it will come to the parietal cells. So gastrin hormone come to the parietal cells and act on CCK receptor and stimulate the acid secretion. Right? So this is the CCK receptor, this one. So this is acetylcholine act on M receptor. Then this is the H receptor. Histamine will act on H receptor, histamine. As I mentioned earlier, histamine is it's not a hormone. It's a substance. It's available in the tissue. So that is, histamine is example for the paracrine type stimulation. That means it diffuse surrounding cells. No need to go to the blood and diffuse in surrounding cells and act on those cells. ළඟපාත තියෙන cells වල act කරනවා. So that is histamine secreted from ELC cells, right? Histamine secreting cells and act on parietal cells by locally diffusion. So histamine, gastrin, and vagus nerve, vagus will stimulate the parietal cells and produce secretion. Right? So this gastrin is coming from G cells. That is the hormone producing cells located in the lower part of the stomach, mucosa in the lower part of the stomach. G cells will come and, G cells will produce gastrin and gastrin will act on parietal cells, right? So this G cells also will be stimulated by vagus nerve. This is the vagus nerve. And neurotransmitter is not acetylcholine, GRP will stimulate the G cells and secrete gastrin. So vagus nerve will stimulate G cells as well as directly parietal cells. The both way it increase the acid secretion. Vagus nerve, parasympathetic impulse carry through the vagus nerve, right? So that is one thing. Then for this gastrin hormone producing G cells also, it has some stimulatory and inhibitory factors. Vagus nerve is stimulatory and it has inhibitory factors also coming from cells called D cells, and this substance will come and inhibit the G cells. Here you can see inhibition. This all GRP is stimulatory and this is inhibition. So this inhibition is operate when the acidity increase. When the H concentration increase, that will directly stimulate D cells, and then D cells will secrete SST or some substance or something and inhibit the G cells. So now you can see when the acid secretion increase, we need to inhibit the parietal cells. So directly we cannot inhibit the parietal cells. We can inhibit the gastric secretion from the G cells. How? Through the D cells producing substance will inhibit the G cells and gastric secretion will be inhibited, right? So now you can see that gastric secretion is very well regulated. If it is increased, it can inhibit the production. If it is required, like vagal stimulation, when the food comes, it increase the production of the HCl, and because HCl is required for the digestion and so on, right? So these are mainly operate in the fundus and upper part of the stomach, and G cells are mainly located in the lower part or antral part of the stomach, right? So that is something you need to understand. Right, okay. Right. So then little bit about the gastrin, as I mentioned earlier. Gastrin is a hormone. Gastrin's acid secretion is stimulated by the gastrin, secreted from G cells and transport from via blood. And gastrin act via CCK-B receptor. Actually, it's very interesting why we call it a CCK receptor. CCK is another hormone important for the pancreatic and gallbladder function. CCK, cholecystokinin. But long time ago, when they found the gastrin, they thought it's a CCK because it's structurally similar, and so therefore they don't know it's a gastrin, and they call it a CCK, and receptor they name as a CCK. But later found that this is not CCK hormone, this is a gastrin hormone. Of course, structural similarity, but it's not exactly the same hormone. So they name it as a gastrin hormone. But still receptor is CCK. හෝමෝන් නම වෙනස් වුණා, receptor ගේ නම වෙනස් වුන්නේ නැහැ. Still we call CCK receptor, gastrin act on CCK receptor, right? So if it easy to remember if it is a gastrin receptor, because gastrin will act on gastrin receptor. But now gastrin is the hormone, receptor is CCK receptor, right? So we can see overlapping action of cholecystokinin and gastrin in the most of the places because they are structurally similar hormone. Gastrin release stimulated by vagus nerve, I already explained in the diagram. Vagus nerve will stimulate by secreting GRP, and also gastrin secretion is stimulated by when we have protein content in the food, or production of protein, digestion of the food. Somehow amino acid, oligopeptide like protein product will stimulate the gastrin secretion. And you can see when we have protein product, we need the acid to digest or activate pepsin. So therefore, gastrin secretion increased by the product of protein. Inhibited by high acidity. I already mentioned that gastrin secretion should not keep it high when the acidity increase. Then it's a destructive. Acid වැඩි වෙනවා නම් gastrin අඩු කරගන්න ඕනේ. We should increase the gastrin. We should decrease the gastrin secretion if there is a acidity. So that is the inhibitory factor. And then I already mentioned D cells produce somatostatin. So that is what I mentioned here. Somatostatin hormone from the D cells. This one. Oh, yes, this is somatostatin. D cells will produce somatostatin, and that will inhibit the G cells, right? B cells and G cells. D cells make a somatostatin produced by G cells, and they act on G cells, right? Right. Okay, right. Right. So those are inhibitory factor for the gastrin hormone secretion, right? So you might confuse by the different name. Gastric secretion mean secretion from the stomach, acid, mucus, and everything, right? Gastrin is a hormone. Gastrin is a hormone. Right? Gastric secretion is something else, right? So in the gastric secretion, mainly we talk about the HCl, but not only HCl, there are some other substance, right? Then this diagram, almost same thing, just to refresh your knowledge or easy to understand. We have parietal cells, 3 receptor. First one, M3 acetylcholine will act. Second one, histamine will act. And third one, gastrin will act. And gastrin will come from G cells through the blood. And G cells can be stimulated by amino acid or digestion of protein, but inhibited by low pH, right? Also, low pH will stimulate D cells. D cells produce somatostatin, and which will inhibit the G cells. This is inhibitor, right? So that is how it will act. So vagus nerve, I already mentioned, vagus nerve act on the G cells through the GRP neurotransmitter and can activate G cells and produce gastrin, right? So that is it. And the same thing, secretin and GIP hormone coming from small intestine can inhibit the acid secretion and can act as a inhibitory factor for the gastrin, right? So you can leisurely try to understand this diagram. I already explained it, but it take time to understand. Don't worry. අවබෝධ කරගන්න පුළුවන් වෙයි. Right. So I already explained inhibitory factors, acid, and something to erase. right? So acid, and these are some sort of inhibitor factor. You can read, I already explained them. And acid secretion steps, probably you can leisurely understand this. I will briefly explain, but you can read them and try to understand. This is already mentioned. H plus will secrete as an active pump, right? So then this H plus coming from bicarbonate and carbon dioxide and water. So carbon dioxide will enter into the cells, and when H plus secreted, bicarbonate molecule will go back to the blood, right? Because this bicarbonate H plus will secrete here, bicarbonate will enter to the circulation. So it's something like that. When we produce more H or more acid, and more bicarbonate will come to our blood, right? මේ bicarbonate එකට යන්න තැනක් තියෙන්න ඕනේ. So H plus will secrete to the lumen and bicarbonate will enter to the blood. So if we produce large amount of acid, more bicarbonate will enter to the our circulation, that we call postprandial alkaline teat, right? Postprandial mean after meal, right? Postprandial is a word used to after meal, and you can see alkaline bicarbonate teat, right? Podi bicarbonate wave එකක් වගේ එනවා if we produce acid in with the meal, right? Then the drug proton pump inhibitors can block this step. So when you get the gastritis, probably you remember you get the drugs like omeprazole, pantoprazole, esomeprazole, like drugs. They will inhibit this proton pump, this mechanism, and prevent the acid secretion, right? I'm sure you or your parents or somebody use this omeprazole or atatatine karla, or pantoprazole, esomeprazole. Those are proton pump inhibitors, right? So this is the process of H plus secretion, and then chloride ion will come to the, according to the concentration gradient, when bicarbonate push back, there's an anion exchange, chloride will come. Chloride then will secrete as a passive process. So finally chloride and HCl secrete, and leisurely try to understand this in the book. Right. So I will stop now. Now it's 45 minutes, and I can continue next lecture, motor function. Before motor function, I will do something about the mucosal barrier in the next day. By the way, what do you have after 10:30? 10:30 වලට මොනවද තියෙන්නේ? කතා කරන්නේ නැනේ. 10:30 වලට මොනවද තියෙන්නේ? Do you have any activity at 10:30?
Yes, sir, we have a public health lecture.
Okay, good. Right. So the next lecture tomorrow, I think, or later, about gastric motility, or not tomorrow, I think Monday or later, I will continue from here. Right. Okay. So I will stop now. So have a nice day.